This lecture continues our series of 3 about energy metabolism. We'll talk about fermentation pathways. At the end of this lecture, you should be able to describe major features of fermentation pathways, including steps to convert monosaccharides to pyruvate, sites of ATP generation, and the importance of substrate level versus electron transport phosphorylation. Your reading is Chapter 14 from the textbook. In an earlier lecture on degradation of feed, we gave a brief overview of glucose fermentation. Recall that during fermentation, glucose is first converted to pyruvate. Then pyruvate is converted to VFA, lactate, carbon dioxide, hydrogen, and methane. This yields energy for the microbe in the form of ATP. For the remainder of the lecture, we'll go over the pathways in fermentation in great detail. The details of biochemical pathways can sometimes be disorienting, but you can put them in the context by going back to this slide. What features do you need to know of these pathways? Do you need to memorize them entirely? No, not in the sense that you have to draw them on a blank sheet of paper. But you should be able to do a couple of things. You should know key points that are written on the left side of the slide. And you should also be able to identify and explain major features of a pathway if one is shown to you on an exam. As we mentioned, in the first step of glucose fermentation, glucose is converted to pyruvate, and we see how this is done with the EMP pathway. In earlier classes, you probably referred to this pathway as just glycolysis. In the first half of this pathway, glucose is phosphorylated twice. First by glucokinase, or PTS, and second by phosphofructokinase, PFK. This represents an initial input of 2 ATP. In the second half of this pathway, the payout phase, 4 ATP are generated. First, phosphoglycerate kinase, PGK, generates 2 ATP. Second, pyruvate kinase, PK, generates another 2 ATP. From the figure, it looks like PGK and PK yield 1 ATP each. That's because, as it's written in the figure, glucose produces only 1 pyruvate. In reality, 2 are produced, and everything in the red box should be multiplied by 2, including the number of ATP produced. The EMP pathway is the predominant pathway for degrading hexoses. Over 90% of glucose is degraded this way. To this point, we've talked about fermentation of glucose. Pentoses are released from hemicellulose, and they're fermented as well. To ferment these, they're first converted to pyruvate, using 2 pathways, either the pentose phosphate or the phosphoketolase pathway. Both of these pathways share the feature that they feed into the EMP pathway. In the pentose phosphate pathway, pentoses are first converted to sugar phosphates by epimerases and kinases. For example, ribose is converted to ribulose 5-phosphate. These sugar phosphates are subsequently converted through a number of intermediates and then fed into the EMP pathway. There are a number of steps involved, and you're not responsible for knowing the details. The pentose phosphate pathway is the predominant one for degradation of xylan found in hemicellulose. A full 75% of the xylan is degraded by this pathway. Xylan enters through xylulose 5-phosphate. The phosphoketolase pathway begins similarly to the pentose phosphate pathway, in that pentoses are converted to their sugar phosphates with epimerases and kinases. These pentose sugars are then cleaved by phosphoketolase, which gives the name of the pathway. Both acetate and glyceraldehyde 3-phosphate are produced, and the latter enters the EMP pathway. Some xylan, 25%, is degraded by this pathway. Less is degraded by this pathway because the pathway yields less ATP than the pentose phosphate pathway. You're not responsible for knowing the exact yield of ATP, just that the phosphoketolase has a lower yield. Once pyruvate is produced, it can be converted to VFA, lactate, carbon dioxide, hydrogen, and methane. The conversion of pyruvate to lactate is the simplest, and this is the classic fermentation pathway that you've seen in many previous classes. It produces no ATP, but it does serve a purpose. It disposes of the reducing equivalent NADH. We'll discuss this concept later, but in short, disposing of NADH prevents it from accumulating and allows fermentation to continue. Another classic fermentation pathway is the conversion of pyruvate to ethanol. This disposes of reducing equivalents without generating ATP, and in that sense is similar to lactate production. This fermentation is not important in the rumen, however. Ethanol is detectable only in trace amounts, less than 0.5 millimolar. Lactate production, by comparison, is more important. The conversion of pyruvate to acetate produces 1 ATP per pyruvate. This is done by substrate-level phosphorylation. This is a mechanism of ATP synthesis in which a phosphorylated substrate transfers its phosphate group to ADP and produces ATP. We see this in the last step, which is mediated by acetate kinase. Acetylphosphate transfers its phosphate group to ADP, forming ATP and acetate. The yield of fermentation to acetate is 4 per glucose when factoring in the EMP pathway. The EMP pathway itself yields 2 ATP per glucose. When pyruvate is fermented to acetate, 1 ATP is yielded per pyruvate, which yields a total of 2 ATP per glucose, since glucose yields 2 pyruvate. The conversion of pyruvate to butyrate produces 1 ATP per 2 pyruvate. It forms ATP by substrate-level phosphorylation, and it can do this in 2 different ways. In the first way, pyruvate is converted to butyryl phosphate, and then butyrate kinase transfers a phosphate from ADP to ATP. This forms 1 ATP and butyrate. The second way is more indirect. Butyryl-CoA is converted to butyrate, but this in itself does not form ATP. Rather, ATP is formed through a series of steps involving phosphotransacetylase and acetate kinase. Butyryl-CoA donates its CoA group to acetate, forming acetyl-CoA. The acetyl-CoA is then converted to acetylphosphate by phosphotransacetylase. Finally, ATP is formed when acetate kinase uses acetylphosphate to phosphorylate ATP. Note that there is no net production of acetate. It is both consumed and produced by this reaction. The total ATP yield when producing butyrate is 3 per glucose when factoring in the EMP pathway. The conversion of pyruvate to propionate can be accomplished by 2 different pathways, the randomizing pathway and the direct reductive one. The randomizing pathway produces about 1 1/3 ATP per 2 pyruvate. This is accomplished by electron transport phosphorylation. That is, ATP synthesis accomplished with an electron transport chain. In the randomizing pathway, electron transport and ATP formation occur when fumarate is reduced to succinate. The figure in your textbook does not show the steps in detail. It simply shows ADP is being phosphorylated to ATP during reduction of fumarate to succinate. To produce ATP by electron transport phosphorylation, fumarate reduction first generates a proton motive force. The figure at right shows you how that proton motive force is generated. The proton motive force is generated by the consumption of 2 protons inside the cell. This follows from the influx of 2 electrons into the cell, as shown in the animation here. We note that there are 2 protons that are consumed per 2 electrons. You'll need to remember this value in order to calculate the ATP yield of fumarate reduction. The proton motive force that we formed drives ATP synthesis by ATP synthase. In the figure at right, we can see that protons flow through ATP synthase and ATP are generated as a result. To calculate ATP yield, we note that 2 electrons enter the cell per fumarate. 2 protons are consumed by 2 electrons, and 1 ATP is generated per 3 protons. Multiplying these values together shows approximately 2/3 of an ATP is formed per fumarate. This means that 1 1/3 ATP are produced per 2-pyruvate, because 2-fumarate lead to 2-pyruvate. These ATP yields are approximate because no one really knows the stoichiometry of the ATP synthase. The figure suggests that 1 ATP is generated per 3 protons, but there's debate over what is the true stoichiometry. Because the randomizing pathway produces 1 1/3 ATP per 2-pyruvate, there's a total yield of 3 1/3 ATP per glucose when factoring in the EMP pathway. The second pathway for producing propionate is a direct reductive pathway. This likely produces no ATP per pyruvate. Some have speculated that ATP could be produced at the outline step by an unknown mechanism. ATP synthesis has not been shown, but the energy change at this step is large enough that it could support ATP synthesis. The low growth yields of organisms with this pathway indeed suggest that no ATP are yielded. If no ATP are yielded, then 2 ATP are formed per glucose when factoring in the EMP pathway, the only source of ATP. We've now finished fermentations that are carried out by bacteria. We'll shift our focus to methanogenesis, which is carried out by methanogens. During methanogenesis, hydrogen and CO2 are converted to methane. 2 pathways can be involved, one pathway without cytochromes and a pathway with cytochromes. The pathway without cytochromes produces about half an ATP per carbon dioxide reduced with hydrogen. As we can see in the red box, the reduction of carbon dioxide generates an electrical chemical potential of sodium. This potential then drives ATP synthesis by ATP synthase. The last step of methanogenesis involves coenzyme M. This step does not generate ATP, but it's of note because analogs of coenzyme M have been developed to inhibit methanogenesis. These analogs include ones like bromoethane sulfonate, and if dosed in vitro, it's very effective in reducing methane production. We'll discuss the use of analogs later in the class. The ATP yield of methanogenesis is not really known, and the value of 1/2 ATP per carbon dioxide is approximate. The second pathway of methanogenesis with cytochromes produces about 1.5 ATP per carbon dioxide. The pathway is similar to that without cytochromes, but more ions, 4 protons, are pumped out by cytochromes, as you can see in the red box. The higher electrochemical potential of protons that results drives the synthesis of more ATP. The ATP yield is not really known, just that it's higher than for the pathway without cytochromes. There is a trade-off between these pathways. The pathway with cytochromes yields more ATP, but the hydrogen concentrations have to be higher. Higher hydrogen concentrations mean that more energy is released during methanogenesis, supporting the synthesis of more ATP. Methanobrevibacter ruminantium, the predominant cultured species, does not have cytochromes. However, methanogens of the order Plasmatales, which are a major uncultured group, may have them. Enrichment experiments show that this group uses methylamines, and all cultured methanogens that use methylamines have cytochromes. We'll end this lecture with a discussion of the importance of substrate level versus electron transport phosphorylation. The pathways presented in this lecture suggest that most ATP is produced by substrate level phosphorylation, and indeed, substrate level phosphorylation was once thought to dominate pathways of anaerobic metabolism. Electron transport phosphorylation was recognized as important only in a few cases, for propionate production and for methanogenesis, as well as a few other pathways. In recent years, though, since the release of your textbook, electron transport phosphorylation has been found to be coupled to many pathways. Ion pumps have been recently discovered, and these generate an electrochemical potential of protons or sodium. This potential, in turn, drives ATP synthesis. Electron transport phosphorylation has been found with butyrate formation from glucose. It can drive an additional 1.5 ATP per glucose. The ion pumps RNF and ECH pump out sodium and protons. This, in turn, forms an electrochemical potential of protons or sodium. This potential can then drive the synthesis of about 1.5 ATP. This mechanism of ATP synthesis is quantitatively important. It increases the ATP yield by up to 50%, from 3 to 4.5 ATP per glucose. The take-home points of this lecture are that glucose is converted to pyruvate primarily by the EMP pathway. Pentoses are converted to pyruvate primarily using the pentose phosphate pathway and EMP. Pyruvate is fermented to VFA and lactate. ATP is generated by both substrate-level phosphorylation and electron transport phosphorylation, with the importance of the latter being increasingly recognized. Finally, some methanogens use cytochromes in the pathway for methane formation. This lecture